Hi, my name is Camille Derderian, and I'm a third-year graduate student at UC San Francisco in Jeremy Reiter's lab. My research interest lies in understanding the intersection between form and function in cell biology. How do cells build and maintain distinct structures that carry out essential signaling functions? In the Reiter lab, we study primary cilia. The primary cilium is an ancient, evolutionarily conserved organelle that serves as a signaling tower in the cell. It performs critical signaling roles throughout embryonic development and adult tissue maintenance, and dysfunction in ciliary signaling results in dire consequences for the health of the organism. The structure of the primary cilium is unique compared to other organelles because it is incompletely membrane-bound. It is contiguous with the plasma membrane at its base, and it is open to the cytoplasm of the cell. Despite this, the primary cilium can remain both compositionally and functionally distinct from the plasma membrane and the cytoplasm. This distinction is achieved by a few means. At the base of the cilium is the mother centriole, called the basal body in the context of the cilium. This structure nucleates the ciliary axoneme, a microtubule-based structure that serves as the tracks upon which cargo can be transported in and out of the cilium. Proteins can embed and enrich specifically in the ciliary membrane and carry out unique signaling functions at this location. But my project focuses on the membrane itself. We are gaining insight into the ways in which lipids that make up the primary ciliary membrane differ in character from those in the plasma membrane. In the plasma membrane of the mammalian cells, there is a common distribution of phospholipids which make up the lipid bilayer. Most abundant are the two neutrally charged phospholipids, phosphatidylcholine and phosphatidylethynolamine. Less abundant, but still critical for cellular function, are phosphatidylinositol, cholesterol, and phosphatidylserine. Phosphatidylserine is the most abundant negatively charged phospholipid in the cell, and it resides exclusively in the leaflet of the lipid bilayer that faces the cytoplasm. To investigate the distribution of phosphatidylserine in ciliated cells, we turned to the tools we had at our disposal. We utilized a biosensor that is able to specifically bind to phosphatidylserine in order to localize phosphatidylserine in whole living cells. Using this tool, we made a fascinating discovery. Phosphatidylserine is highly localized to the primary cilium. Given this information, we adapted a tool to locally manipulate phosphatidylserine at the ciliary membrane. This tool utilizes an essential enzyme that converts phosphatidylserine into phosphatidylethynolamine, effectively removing phosphatidylserine from the ciliary membrane. When we express this tool in cells, we can see that phosphatidylserine is depleted upon delivery of our tool to the cilium. This local manipulation allows us unique access to understanding the function of phosphatidylserine at this specific location. With this novel tool, we will investigate how lipid content can change when phosphatidylserine is removed from the primary cilium. We can also look at membrane protein localization, as well as look in vivo in zebrafish models to identify how changing phosphatidylserine localization in zebrafish cilia may affect their development. I look forward to pursuing these fascinating questions, and I want to extend such gratitude to the ARCS Foundation for their generous support of my work. Thank you.